Welcome to Fort Lewis College. We're the P3 EPA group, and we're working to develop a field-deployable digital droplet PCR system for the rapid detection of waterborne bacteria pathogens. Over to Caitlin, and she's going to show you where we get our bacteria.
Hi everyone, my name is Caitlin, and I'm going to be showing you how I collect and process our water samples. I first go to a local stream where I know there to be E. coli in the water samples and collect following EPA methods. I then bring those water samples back to the lab and filter them through these filtration units. 100 milliliters of water sample is put into this upper receiver and filtered through. All of the bacteria is collected on one of these filter plates. These filter plates are then put into a second filtration unit, flipped over, and then flushed using a backwash flushing technique. All of the water collected and the bacteria encapsulated in the filtration fluid is then recovered from this lower receiver and is then sent off into a next process so it can be counted. All of these methods are done following standard EPA guidelines for filtration and water collection. I'd now like to hand it over to Nick to say some more about our photolithography processes.
Hi, I'm Nick. Photolithography is the process of transferring micro and nanoscale patterns onto a photosensitive material. The fabrication of microfluidic devices uses a liquid photoresist called SU8 to form the patterns that mold the final PDMS channels. The photolithography process is composed of three main steps: spin coating, UV exposure, and development. These patterns are then molded with PDMS, and the PDMS is bonded to a microscope slide. On to Jesse to discuss how these chips are used.
Hi, I'm Jesse. So once the microfluidic chips are made, we run them over here to the syringe pumps. These pump fluids at a specific rate, which allows us to create microfluidic droplets which house one cell of bacteria. The surfactant over here runs as a pincher to the PCR product, which gets chopped up into little tiny droplets. Those droplets are sent over to the thermocycler, which will amplify the DNA. And to tell you more about the thermocycler is James.
Hi, my name is James. This is our FLC PCR system for thermocycling microfluidic droplets. PCR is performed by first raising the DNA to a high temperature to split the DNA in half. Then the temperature is lowered, allowing the DNA strands to reform, thus duplicating them. Do this many times and you end up with millions of strands of DNA. This system uses an ATmega 328P and thermoelectric peltiers to manage the temperature. This is all managed by a Raspberry Pi running a touchscreen interface developed in C++ and SDL2. Now back to Jesse for bacteria counting.
Finishing it off, we bring our newly fluorescing microfluidic droplets to our optical setup. Here we use a blue light laser, which reflects one way off of a dichroic mirror onto a channel which houses our droplets. Those droplets, when hit by the light, fluoresce back green. That travels through our optical setup and a bandpass filter, which only allows green light to pass, into our photomultiplier tube, or PMT, which counts the number of photons that hit it and outputs the voltage. So from beginning to end, we go from bacteria in the water to voltages on a computer. That's how we get our bacteria counts. Thank you for your time.